GMO modified organisms. Contents. What are genetically modified organisms? Production of genetically modified organisms. Principal objective of production. Existing models. Genetic engineering with traditional breeding. Pros and cons. Need of DAR. What are genetically modified organisms? A genetically modified organism or GMO is an organism whose genetic material has been altered using genetic engineering techniques. Organisms that have been genetically modified include microorganisms such as bacteria and yeast, plants, fish, and mammals. Source of genetically modified foods and are also widely used in scientific research and to produce useful goods other than food. Production of genetically modified organism. Genetic modification involves the insertion of deletion of genes. When genes are inserted, they usually come from a different species, which is a form of horizontal gene transfer. In nature, this can occur when exogenous DNA penetrates the cell membrane for any reason. To do this, artificially may require attaching the genes to a virus or just physically inserting the extra DNA into the nucleus of the intended host with a very small syringe or with very small particles fired from a gene gun. Agrobacterium's ability to transfer genetic material to plants, or the ability of lentiviruses to transfer genes to animal cells are natural examples of gene transfer. Principal objective of production. The basic principle for producing a GMO was to add new genetic material into an organism's genome. This is called genetic engineering and was made possible through the discovery of DNA and the creation of the first recombinant DNA molecules by Paul Berg in 1972. Transgenic plants. Transgenic plants have been engineered for scientific research to create new colors in flowers and to create improved crops. In research, plants are engineered to help discover the function of certain genes. One way to do this is to knock out the gene of interest and see what phenotype develops. Another strategy is to attach the gene to a strong promoter and see what happens when it is overexpressed. GM crops. In agriculture, genetically engineered crops are created to possess several desirable traits, such as resistance to pests, herbicides, or harsh environmental conditions, improve product shelf life, increase nutritional value, or production of valuable goods such as drugs. Plants. Plants including algae, jatropha, maize, and other plants have been genetically modified for use in producing fuel known as biofuel. Microbes. Bacteria were the first organism to be modified in the laboratory due to their simple genetics. These organisms are now used for several purposes and are particularly important in producing large amounts of pure human proteins for use in medicine. Genetically modified bacteria are used to produce the protein insulin to treat diabetes. Similar bacteria have been used to produce clotting factors to treat hemophilia and human growth. Hormone to treat various forms of dwarfism. Mammals. Ralph L. Brinster and Richard Palmiter developed the techniques responsible for transgenic mice, rats, rabbits, sheep, and pigs in the early 1980s. They established many of the transgenic models of human disease include the first carcinoma caused by a transgene. The process of genetically engineering animals is a slow, tedious, and expensive process. However, new technologies are making genetic modifications easier and more precise. Intended purpose of genetic modification of mammals: to research human disease, for example, to develop animal models for this disease; to produce industrial or consumer products. The example is fibres for multiple uses; to uh, produce products intended for human therapeutic use; to enrich or enhance the animal's interactions with humans; to enhance production or food quality traits; to improve animal health. GM fish are used for research, and pets are being considered for use as foods as aquatic pollution sensors. Genetically engineered fish are widely used in basic research in genetic and development. Two species of fish, zebrafish and medaka, are most commonly modified because they have optically clear chorions, rapidly develop, and the one-cell embryo is easy to see and micro-inject with transgenic. The glowfish is a potent brand of genetically modified fluorescent zebrafish with bright red, green, and orange fluorescent color. Although not originally developed for the ornamental fish trade, it became the first genetically modified animal to become publicly available as a pet when it was introduced for sale in 2003. They were quickly banned for sale in California on the grounds of ethical issues. Breeders sometimes have to cross many plants over multiple generations to produce the desired trait. GM techniques allow new traits to be introduced one at a time without complications from extra genes and extensive crossbreeding. GM techniques also allow traits from different organisms to be applied, such as pest resistance. Pros of genetic engineering: production of human insulin. Patients suffering from diabetes are not capable of producing enough insulin, so there arises a need for such people to obtain insulin from external sources. With the help of genetic engineering, human genes can be transferred into other mammals for the production of insulin. The mammals, like sheep and goat, are used as medium, with human genes playing the role of software or the brain, containing necessary genetic information for produce insulin. The GMOs, like some viruses, are used in gene therapy. Gene therapy can be used in the treatment of various genetic disorders and diseases like sickle cell anemia, muscular dystrophy, and cystic fibrosis. Creation of new organs. The unavailability of organs for transplants is a big problem today. The creation of new organs in order to increase the supply of desired organ is possible by means of genetic engineering. The regeneration of new tissues is carried out by the injection of a growth factor using a tissue injector. Another procedure for creating new organs, in which a scaffold made from biodegradable biodegradable polymers is used to contain the plant cell. This scaffold is placed in a position where the new growth is expected. Eventually, the scaffold breaks down or dissolves, and a completely new organ is formed. Usage in agriculture. Genetically modified plants have many applications in the field of agriculture. Genetic modification or engineering is used for increasing the production of crops, pest control, weed management, etc. the genetically modified foods are also produced to make them more nutritive. For example, the incorporation of digestible iron in the genetically modified crops influence health in a positive way. Cons of genetic engineering. Main argument made against activity genetic modification is that it leads to unpredictable outcomes or side effects. Genetically modification is considered to be unnatural and thus unfit in the, in the context of natural ways like breeding or crossing in the plants and animals for bringing out the best in them. Thus, the possibility of unpredictable alteration taking place in the genetic makeup of organisms is one of the biggest cause of worries among scientists regarding the whole issue of genetic modification. It is important to note that FDA has not approved consuming animals that are genetically modified. The stand taken by FDA implies that they don't want these genetically organisms to become a part of the food chain. Harmful effect on crops. The genetically modified crops which the farmers plant in their fields have the same genetic makeup. Cross-pollination of such plants with other plants increase the risk of contamination. The Bt or Bacillus thuringiensis genes present in the GM crops kill the insect like bees, ladybird, beetles, butterflies, etc. Thus, health organisms too are affected along with pests. Risk of misuse. The risk of the information regarding these techniques falling into wrong hands should be considered while studying the pros and cons of genetic engineering. Mindless cloning of animals and plants for commercial purposes would definitely have adverse effects on the gene pool we have today. The process of genetic erosion might be accelerated with increase in the number of genetically modified organisms. Some facts: GM seed is taking over our natural food supply. GM plants proven to be poisonous, killing insects, birds, butterflies, and destroying our ecosystem and slowly killing animals. Scientists of FDA have reported that the process of genetic engineering and traditional breeding are different and they lead to different risks. GM food contaminates common garden chemicals are also killing our bees to protect ourselves.